were in the region of Israel in the Roman period, but we don't know historically if that's true or not. What we do know is that the first Ashkenazi communities appeared in this region of Ashkenaz, which is around the border of Germany and France. So these first Jewish communities appeared in this region around the end of the 9th century, which is relatively recent. We're talking about something like 1,000 years ago. And we also know that later they expanded rapidly in Eastern Europe. But there's a lot of open questions about this early history, about what happened there. For example, we don't know who were the first Ashkenazi Jews, where did they come from to the region of Ashkenaz. We don't know when did they move to the east. Was it a subgroup of these communities in Ashkenaz, or were they different Jews over there? And we don't know when Jews intermarry with others. Like, we know that they did by their DNA, but we don't know when did it happen. So there's a lot of open questions about the details of the formation of this population. And there have been a lot of studies in the field of population genetics about the population of Ashkenazi Jews, mainly because of the high burden of disease mutations in this population. And one finding that was replicated in a lot of the studies is, okay, I'll explain what do we see here. So this plot called PCA plot, and PCA is a method that we use to represent the differences between genomes of different individuals on a two-dimensional plot. So each point that you see here in the plot is representing one individual, the genome of one individual. So, okay, just keep in mind that I will walk you through this plot, but the purpose of this plot is to compare the genomes of present-day Ashkenazi Jews to genomes of Europeans and Middle Easterners. So all of the individuals in this plot are present-day individuals, and the gray points represent Middle Easterners and Europeans. So if two points are closer to each other, it means that their genomes are more similar to each other, and if they are distant, it means that they are more different than each other. So you can see this Middle Eastern axis, starting from the Caucasus, then Iranian, Lebanese, and until Bedouin in the right corner. Okay, so each group cluster together because they are more similar to each other. And then they created this Middle Eastern axis. Okay, and again, everything is based only on genetics. I just added the labels so you will understand what we see here. You can also see European axis, starting from Eastern Europe. They are the most different from the Middle Easterners. Then we move to Western Europeans, and then in blue you can see different Southern European populations. Okay, so the Southern Europeans are the European populations that are the closest to the Middle Eastern populations from genetic point of view. A few more. Okay. So in green you can see Ashkenazi Jews, and you can see that they're in the middle between the Europeans and the Middle Easterners. Okay, so this finding was replicated in all kinds of ways in different studies. Ashkenazi Jews in their gene pool have ancestries of Middle Easterners and also ancestry of Europeans. So that means that even if they came from the Middle East originally, at some point they mixed with some local non-Jewish Europeans. And you can click. Okay, and in pink you can also see other Jewish groups. So you can see that Turkish Jews, which represent Sephardic Jews, are very close to Ashkenazi Jews, and also North African Jews. And you can also see some Middle Easterner Jews, if you can, yeah. So you can see Iraqi and Iranian Jews on the Middle Eastern ancestry axis, a little more far from the Ashkenazi and Sephardic Jews. Okay, so that is one finding. Another thing that was found about the Ashkenazi Jews from genetic studies is that there was a very strong founder event or bottleneck in the early history of this population. So what does it mean, founder event or bottleneck? So let's assume that we have this box of balls in all kinds of colors, and then we choose randomly 4 balls out of this box, and these 4 balls randomly have these 3 colors. And then we created this new box of balls from these 4 balls. So the new box has only 3 colors. So we lost a lot of the diversity of the colors that we had in the original box, in the original population, and now we have much more homogeneous box. So that's exactly what happened in the history of Ashkenazi Jews. They had a very strong founder event, which means that a very small group of founders disconnected from the original population and created the current population of Ashkenazi Jews. And all of the present-day population descended from this very small group of founders. That's why the Ashkenazi population is very, very homogeneous from a genetic perspective. And so there were all kinds of proofs for this founder event. And I'm sure a lot of you heard about the high frequency of disease mutations in Ashkenazi Jews. So this is a result of this bottleneck, because the small group of founders had some random disease mutations like all of us have, but then because the whole population descended from this small group, a lot of people in the population carried the mutations that the founders had. Okay, so that's the reason there's a lot of disease mutations in this population. We also see a long chunks of DNA that are identical between unrelated Ashkenazi Jews, and again, this is a result of the bottleneck, of the founder event. And a previous study of Shai Karmi, my PhD advisor, estimated the bottleneck parameters. So they estimated that the bottleneck happened 700 years ago, and that it was so strong that the effective population size during the bottleneck was around 300. So these are estimations. We are not sure if that's the right numbers, but we are sure that there was a bottleneck. Okay, and the population today is very large, around 10 million individuals, and as I said, it's genetically very, very homogeneous. It's very striking about the Ashkenazi Jewish population. So you can see here in the plot, this is a PCA, but this time only of Ashkenazi Jews, and they are colored by the country of origin in Europe. And you can see that regardless of the country of origin, they all created this uniform cloud. Can you hear me like this?
Check.
Okay, thank you. So you can see that regardless of the country of origin, they create this uniform cloud. So they are very, very similar genetically, regardless of the country of origin in Europe. And the population is so... Okay. Should I keep talking like this? Yeah, okay. So the population is so homogeneous that if we take two random Ashkenazi Jews that are unrelated, they will look genetically like third or fourth cousins. So very similar. And okay, so there were a lot of studies about present-day Ashkenazi Jews trying to understand their history, but we ask ourselves, can we use ancient DNA of individuals that live closer in time to the time of the formation of this population to get a better understanding of the early history and the origins of Ashkenazi Jews? And that's where our study begins, in a city called Erfurt in Germany. So this is a map of Germany, and the blue circle is where Erfurt is. So Erfurt was a city in the region of Ashkenaz, close to the border of the region from the east, but inside this region of Ashkenaz. And there are records of Jewish settlement in Erfurt starting from the late 11th century. But then in 1349 there was a wave of pogroms in the area, and most of the Jews in Erfurt were killed, and the community was completely destroyed. But then only 5 years later, thank you. So only 5 years later, the city invited Jews to come back to Erfurt, and they did come back and founded a second Jewish community in Erfurt. And this community was successful and became one of the largest communities in the region. But then only a century later, all Jews were expelled from Erfurt, and that was the end of Jewish presence in Erfurt until the 19th century. And you can click another one, thank you. So this is the old synagogue in Erfurt. This is the oldest synagogue in Erfurt, Europe, that is still standing. And there is also the famous Erfurt treasure that was like some Jews before the pogrom hide this treasure in the wall of one of the houses, and it was found like 25 years ago. And it is presented in the old Jewish synagogue in Erfurt. Okay, so during all of this time of the medieval Jewish communities in Erfurt, Jews buried their dead in the same cemetery, the medieval Jewish cemetery in Erfurt. And after the Jews were expelled from Erfurt, a granary was built on top of the Jewish cemetery. And this granary just stood there until something like a decade ago. They decided to convert this granary into a parking lot. Yeah. So in order to do that, they wanted, thank you. So this is the granary, what you see here. And they wanted to build a ramp for the cars in front of the granary. That's like the ramp that you can see here. So they needed to dig in the ground in front of the granary, and there are still Jewish skeletons in the ground. So they did a rescue excavation for this small region and move the skeletons from there to a modern Jewish cemetery. Okay, so most of the skeletons are still in the ground where the granary was, but this like small section they excavated in order to move the skeletons. So that's how we got approval to use teeth, detached teeth from these skeletons to do a DNA research. Okay, because teeth are not exactly considered as part of the body in Judaism. You don't need to bury a tooth when it falls. So we got the permission to use teeth. Sorry, you can. Thank you. You can also. Yeah. So we got the permission to use the teeth to do a DNA research, and eventually the skeletons were reburied in 2021. Yeah, you can do another 2 clicks. Yeah. Okay. So we got 33 teeth. 33 teeth of 33 different individuals. We had 19 females and 14 males. We detected 2 families of first-degree relatives in our samples. So 1 family of mother and 2 children, and 1 family of father and a daughter. And a large share of our samples were kids under the age of 20 when they died. We couldn't identify the cause of death. There were no signs for violence on their skeletons, except for 1 individual that was killed by a sword. So we assumed that they were not victims of the pogrom. We also didn't. So at the time that they died, it was the time of the Black Death in the region, but we couldn't find any traces of the Black Death bacteria, so we also assumed that they did not die from the plague. We did radiocarbon dating for some of the samples. We wanted to understand if they belong to the first community before the pogrom or the second community, but unfortunately the time range of the radiocarbon dating overlapped with the end of the first community and the beginning of the second community, so we couldn't use it to distinguish between the 2 different communities. But according to some archaeological findings, it is more likely that the samples that we studied belong to the second community that was founded after the pogrom. Okay, so now we can talk about the results. And another one. Thank you. So this is the same PCA that I showed you a few slides ago. And now onto this PCA, we projected the Erfurt samples. Okay, we wanted to see if they are similar to present-day Ashkenazi Jews or something else. So you can see the samples here in red, and you can see that they cluster pretty nicely with the modern Ashkenazi Jews in green. So they are very similar genetically to modern Ashkenazi Jews. And we did some simulations to understand what could be the maximal gene flow from non-Jewish populations into the Ashkenazi population since the time of Erfurt to today. And we estimated in maximal 2 to 4% of gene flow from non-Jewish European populations into the Ashkenazi population in the past 700 years. So it means that it's very, very low level of gene flow from other populations. It represents very high level of endogamy in this population in the past 700 years. It doesn't mean that Jews didn't intermarry with non-Jews. It means that Jews that married non-Jews probably were not buried in the Jewish cemetery or did not continue to be part of the Jewish community. So it means that the massive admixture between Jews and non-Jews that we know that happened by the European ancestry in the Ashkenazi DNA. we know it happened, it must have been happened before the time of Erfurt, before the 14th century. Okay, so that's one thing that we can see from this PCA. But another thing, if we do zoom in into this Erfurt and modern Ashkenazi Jews individuals, is you can see that the Erfurt samples are much more diverse than the present-day Ashkenazi Jews. The present-day Ashkenazi Jews, the present-day Ashkenazi Jews form this tight cluster in the middle, and the Erfurt Jews spread from these two sides of the present-day Ashkenazi Jews. And you can think that maybe it's because our sample size of the present-day Ashkenazi Jews is very small, but even when we added 700 present-day Ashkenazi Jews, that's the next slide. Thank you. So even when we added 700 Ashkenazi Jews, that's the green cloud you see here, they still form this tight cluster in the middle, and the 33 individuals from one city in Germany in medieval times were much more diverse than the present-day population. And so we asked ourselves whether we see here one very diverse group, or maybe we have here two or more groups in the Erfurt samples. So, because we were very surprised by this finding. So we did all kinds of statistical modeling and analysis, and we concluded that indeed we have here two different groups. So we called the first one Erfurt, sorry, not yet. We called the first one Erfurt EU, because these samples were closer to the European populations on the PCA, and the second one Erfurt ME, because these samples were closer to the Middle Eastern populations on the PCA. And then we started to compare between these two groups to understand what's the differences, and we found that the Erfurt EU group had much higher affinity to Eastern European populations. And when we modeled the ancestry of each individual from the Erfurt samples as a mixture of Eastern European, Southern European, and Middle Eastern ancestries, so that's what you see here. We used, to do this modeling, we used proxies of Russian as Eastern Europeans, Lebanese as Middle Easterners, and South Italians as representing Southern Europeans. So we used all of these present-day individuals to model the ancestry of the Erfurt individuals. And each bar in this plot represents the ancestries of one individual, and the different colors represent the different ancestry components. So in red, you can see the Eastern European ancestry, and you can see that all of the Erfurt EU individuals have substantial Eastern European ancestry that is missing in most of the Erfurt ME samples. Okay, so we have here one group that had some probably recent admixture with Eastern Europeans compared to the other group. And another thing you can see is that all of the samples, you can see it in the blue component, all of the samples has a substantial Southern European ancestry. So this is consistent with one of the theories about the origins of Ashkenazi Jews that claims that they arrived from Southern Europe and maybe specifically Southern Italy, where there was a very well-established Jewish community at the time. So this is consistent with this theory, but we should be very cautious about the interpretation of these models, because as I mentioned, we use modern individuals as a proxy to the sources of these ancestries. So we used modern South Italians that, for example, have also some Middle Eastern ancestry. Okay, so it could be that this Southern European component that we see also represents some Middle Eastern ancestry within it. So it's consistent, but it's not a proof that that's what actually happened. Okay, so we can move on to the next slide. Another one. Okay, another cool thing that we can do with ancient remains, beside the DNA part, is to detect the level of different isotopes in the teeth, and the level of different isotopes can indicate something about the environment when the tooth was formed. So if different individuals have different levels of these isotopes in their teeth, it probably indicates that they were born in different places, okay, with different type of food and different environment. So we did it for the Erfurt samples. We wanted to see if the Erfurt EU and Erfurt EU and Erfurt ME have a different level of isotopes in their teeth. And we did find that when we measured the levels of oxygen and carbon in the teeth, we did find differences between Erfurt ME and Erfurt EU. Erfurt ME is the, Erfurt ME is the blue points, and Erfurt EU is the red points. You can see they have some different levels of these isotopes, and that probably means that they had some different geographic origins, and some of them were not born in Erfurt, even though they were buried over there. Okay, so if we rethink about the model of the history of the early history of Ashkenazi Jews based on our findings, so it is consistent with some origins in Southern Europe. So maybe there were some immigrants from the Middle East to Italy in the times of the Roman period, and then at some point subgroup of these Jews moved to the region of Ashkenaz. And we also detected these two different Jewish groups in one cemetery. So we know based on historical records that at the time, at the medieval times, there were two culturally different Jewish groups at the region. But until today, we didn't have any reason to assume that they were genetically different. But now, based on our findings, we think that they may represent, these Erfurt EU and Erfurt ME groups may represent these two different culturally groups that were dominant in the medieval times. We know from historical records that there were immigrants from the east of, like outside of Ashkenaz, into Erfurt, into the second community of Erfurt. So we think that maybe this Erfurt EU group that has this Eastern European component represent these Eastern Jews, that some of them arrived into Erfurt, and Erfurt specifically mentioned in some documents as a border city between these two different Jewish groups. So it makes sense that even though we have samples only from one site, we still captured these two different Jewish groups that were apparently genetically different. And when we modeled the present-day Ashkenazi Jews as a mixture of these two groups, we saw that we can model the present-day Ashkenazi Jews as a mixture of these two different Jewish groups, a nearly even mixture between these two different groups. Okay, so that's about the ancestry. Another thing that we wanted to study using our samples is the founder event. First, we wanted to understand whether the Erfurt samples already post-dated the bottleneck. Okay, because we don't have a certain estimate of when exactly the bottleneck happened. We don't know historically what was this event. So we thought that if we will see that the Erfurt samples already experienced the bottleneck, we can say for sure that the bottleneck, this event, happened before the 14th century. So to do that, first we used mitochondrial DNA, which is a type of DNA that is different from the DNA I talked about until now. It's a segments of DNA that are inherited from mother to offspring. So, and Ashkenazi Jews, because of the founder event, so 40% of Ashkenazi Jews has 4 specific types of this mitochondrial DNA. So if you have one of these 4 types of mitochondrial DNA, you probably have some Ashkenazi Jews ancestors. So we wanted to see if these samples from Erfurt have these same types of mitochondrial DNA, and we did find that a lot of them had these types of mitochondrial DNA, even more than present-day Ashkenazi Jews. So that was a very strong signal that these samples already experienced the bottleneck. That's why they have this kind of mitochondrial DNA. And another thing so we did all kinds of analysis that I won't describe now, but another thing that we did was to check whether the Erfurt samples has the disease mutations that are present in modern day Ashkenazi Jews. And so, thank you. So we detected multiple disease mutations in these samples from Erfurt, and you can see here, for example, the BRCA1 gene that is responsible for the increase in chances of breast and ovarian cancers in Ashkenazi Jews that we detected in one of the samples in Erfurt, and another variant that are common in present-day Ashkenazi Jews. We found them back then in the 14th century. So we concluded that indeed these samples post-dated the bottleneck, and the bottleneck must have happened before the 14th century. So now that we concluded that they post-dated the bottleneck, we wanted to use these samples together with the present-day samples we have to estimate the bottleneck parameters, to understand when did it happen and what was the population size at the time of the bottleneck. And the major thing that we found was that the Erfurt samples represent a population that experienced much stronger bottleneck than the bottleneck that is represented by present-day Ashkenazi Jews. So we suggested a different model that you can see here. Actually, the details of the model are not very important, but what's important is that we realized that we shouldn't think about the founder event of Ashkenazi Jews as one historical event that happened to all Ashkenazi Jews at the time, but rather we should think about this founder event or bottleneck as different medieval Ashkenazi groups that experienced different bottlenecks in different intensities, and later all of these groups mixed to form the present-day population. But that the present-day population represents this average bottleneck. But there were, at the medieval times, probably there were some populations that experienced stronger bottlenecks, like the Erfurt population, and some groups that experienced weaker bottleneck. So we came up with this model where there was some ancestral population, then 46 generation ago there was a split and the bottleneck started, for one population that is represented by Erfurt, there was a very strong population, there was a very strong founder event, and for another group there was a weak founder event, and then these two groups mixed and formed the current, the present day population. But that's only like one model. There are a lot of possible models. The main idea is that there were different bottleneck intensities for different medieval groups. Okay, so just to conclude what we found out. So first of all, we saw that there is a little change in the broad ancestry of Ashkenazi Jews in the past 700 years. The Erfurt samples and the present-day Ashkenazi Jews look very, very similar to each other from genetic point of view. And we show that there is a substantial Southern European ancestry component in these samples from Erfurt, with the caveats I mentioned. We detected two subgroups of medieval Ashkenazi Jews, unlike the very homogeneous present-day population, and they may represent the Western and Eastern Jewish groups at the time. And modern Ashkenazi Jews are a mixture of these different Jewish groups. We also thought that the Erfurt population already experienced the founder event, so it must have happened before the 14th century, and that some of the well-known pathogenic variants that are common in present-day Ashkenazi Jews were already present in the medieval samples. Okay, so for those of you who are interested in more details, we have a publication in Cell about these findings, and that's it. Thank you.